This is the last of the set of 3 videos on the statistical tests needed for AQA A-level biology. So the last one we're going to look at is Spearman Rank, otherwise known as Spearman Rank Correlation Coefficient. So the idea of Spearman Rank is that you are going to be looking at an association or a correlation. Okay, so we're going to be looking at 2 continuous variables to see whether they are associated with each other. So, for example, if I have 2 sets of continuous data and I plot a scatter graph, you can see straight away about correlation. So you can see straight away that the one on the right-hand side over here, there's no correlation at all. Don't really need to do a statistical test. We can see that for certain. The one over here on the left-hand side, that has got a positive correlation. And the one in the middle has got a negative correlation. The question is it a significant correlation or is it just due to chance? And that's the job of the Spearman Rank. Spearman Rank tests whether this correlation, this positive or negative correlation, is significant. So let's imagine we do an experiment of height of plants against light intensity. So our null hypothesis is that there is no significant correlation between the height and light intensity. So again, null means no. So we always start with that idea that there is no significant correlation. But if you plot it, obviously there looks like there is some form of positive correlation. We could, try and draw a line, a best fit line going through that. And it does look like there is a positive correlation. As light intensity increases, height increases. But whether it's significant or not, we need to test using statistical tests. In this particular case, we look at Spearman Rank. So correlation coefficient or Spearman Rank, it's the same thing. So again, you will not be required to do this in an exam, but you may be required to interpret it. So we do the statistical test. We then calculate our degrees of freedom. The degrees of freedom are the number of pairs, which in this particular case was 11. And then we go to our critical value table. So here are 11 degrees of freedom. Remember, we always start with a 0.05, which is the 5% probability. And we've got a critical value of 0.536. Now, again, that is the smallest value that our calculation needs to be in order for the correlation to be significant. So as long as our value is bigger than that, then we'll be fine. So if our value said, for example, let's say our value for Spearman Rank was 0.601, then 0.601 would be bigger than 0.536. So I could safely say that the correlation is significant. I can reject my null hypothesis, and there is less than 5% probability that the correlation is due to chance. And like before, this could be written simply as P is less than 0.05. However, it is possible that I could end up with a value of, for example, let's say I calculated it and I ended up with a value for my Spearman Rank of 0.42. That is less than 0.536, and therefore I would say there is no significant correlation. I need to accept my null, and there is more than 5% probability that the correlation is due to chance, and that would be written as P is greater than 0.05. So either possibilities that you could actually have, you need to consider what your value is and compare it. Now, again, often in the exam, they give you the p-value rather than actually giving you the value. Now, just bear in mind that if you're doing a negative correlation, then just make everything negative. So you've got minus 0.536, your Spearman Rank calculation would also be negative. So you're saying, is it more negative than the negative value you've got? Okay, so basically, is it closer to -1 if you're dealing with a negative correlation? With a positive correlation, is your value closer to +1? So let's look at an example. A scientist investigated the effects of changes in temperature and rainfall on the yield of maize, soybean, sugar beet, and sunflower. The scientists calculated the test statistics and found the p-values. They're looking at the effects of crop yield and rainfall or crop yield and temperature, both of which are continuous variables, and they're trying to test whether their association is significant or not. We're not worried about whether it's a positive or negative association in this particular question. It doesn't ask us. All it gives us is the p-values. So what do you notice? What do you need to look for? So in any p-value question, same as with the t-test and the chi-squared test, you want to look to see whether is p less than 0.05, because that is significant. If p is less than 0.01, that's even more significant, because that's less than 1%. If p is greater than 0.05, that is not significant. So I would look at this table and I would just jot down, even before I got to the exam questions, what we're seeing. So we can see straight away that that one is very significant. So I'll call it VS. That one is also very significant because 0.01 is significant, not quite as significant as less than 0.01. So we'll just write it as significant for this particular purpose. Okay, significant. 0.69 is greater than 0.05, so that is not significant. Neither is 0.06, because that's also bigger than 0.05. These two, on the other hand, are very significant because they're less than 0.01. That one is also very significant, and that one is just significant. So what can we say? Well, it looks to me like soybean, there is not a significant correlation between rainfall and crop yield, or between temperature and crop yield. In other words, there is more than 5% probability that correlation is due to chance. However, with the other three, there is a significant correlation between rainfall and crop yield, and between temperature and crop yield. There is less than 5% probability that the correlation is due to chance. And in the case of rainfall and crop yield, all the results of maize, sugar beet, and sunflower are very significant in terms of the correlation. And sugar beet and temperature is also very significant. So this tells you the information straight away. You need to look at the p-values that you are being given.